Swayam Prabha. Digital India. Educated India.
Welcome everybody. So we have been discussing our short-term plasticity, and the last class we had covered the particular model of showing or depression in synapses. So let us just recollect what we had. So in the presynaptic terminal, if we draw like this, then we have vesicles that are there, and we call these as the recovered pool of vesicles. Then there is some that is in the effective pool. So this is actually neurotransmitters in the recovered pool. This is the effective pool, and then there is some inactivated or ineffective pool that is not being used by the postsynaptic side, that is endocytosed and carried back into the endosome, and then we have new vesicles being packed with neurotransmitter. So essentially we had the three states, that is the recovered state, the effective state, and the ineffective or inactive state. So, and with three differential equations, we could describe how the receptor, how the neurotransmitters would be in the different states if we have activity, and how with more activity this kind of a model where we have DRDT, DEDT and DIDT show the behavior that we see with depression, synaptic depression, and that is where we have with a spike in the presynaptic side, with every subsequent spike we see decrease in the size of synapses until saturation, until an asymptotic size is reached. However, this particular model where we have three time constants, that is the effect, that is the recovery to effective, that tau Re, which is very small, of the order of 1 milliseconds. That is, we have tau Re here, and it is taking from the recovered state out into the effective state only when there is a spike. We mark that with a U, or this can also be written as a delta function, where or summation delta function, where each Tj is the spike time, and we will assume that the delta function is 1, or it is not a direct delta function, it is 1 over a narrow period around Tj or just after Tj, and R is being replenished by from the inactive state with a time constant tau IR, and this tau IR, the time constant here, is extremely large, of the order of seconds or 1000 milliseconds. So this discrepancy in the time constants, and here this time constant is tau I, which is of the order of tens or 100 milliseconds. It is this discrepancy in the time constants between the recovered to effective and the I to R, that is tau IR. These two determine whether basically based on activity, if we have more and more activity, we will have depression. It is pretty easy to show, so now we move to the other aspect, which is short term facilitation. So in order to explain short term facilitation, And also in the same kind of model, if we need to represent depression as well, then we have to modify this model in a particular manner. So, if we think about it, let us say that n over a period of time as a function of time is the neurotransmitter pool, which was the entire neurotransmitter pool. So, and so sum of it is, that is in the releasable region. So, this is the entire neurotransmitter pool that is releasable, and so in a simpler model, 1 minus n t is the pool that is driving the process to go towards into n t, that is from non-releasable pool to the releasable pool. So, in our earlier 3-state model, this particular state was divided into 2 parts. One is the effective part and the other is the ineffective or inactive part. So, we will now just for the sake of simplicity, we will combine these two and keep it in as one pool and consider only a single time constant of recovery, that is tau r. However, there is nothing that will stop us from including the ineffective and effective pool also in whatever we are talking about or whatever model we are saying. So, that can still be done. So, but just for simplicity, we are taking a 2-state model, that is the releasable pool and the non-releasable, that is it is not in the pool that is going to be released, and it can be in effective or ineffective state. So, essentially the same model becomes d n t dt equals 1 minus n t, and this is what is driving the tau recovery into to go into the releasable pool. And our usage is based on, let us say, summation delta t minus tj, that is from the releasable pool it is going into the effective into the non-releasable pool, and that is this multiplied by nt. So, this also will be showing depression if we make tau r large, and I mean kind of like depression, but in order to bring in the facilitation aspect, as we have discussed, the facilitation is brought about by one of the important hypothesis is the build up of calcium, and there are lots of experiments in support of build up of calcium that leads to facilitation. And we will say, so basically we need the internal calcium concentration to come into play in this whole picture in the presynaptic side. So, internal calcium concentration in the presynaptic side. And this is going to be proportional to the probability of release. So, the probability of release in the terminal can be a proxy for the calcium, and we can simply put a probability of release in front of the NT. So, depending on so this is this probability of release, the way we have written here, is not going to lead to facilitation. What we need to do is make this probability of release as a function of time. So, that is essentially PT. And for the facilitation, the idea is that there is, for every spike, there is some leftover calcium that is going to be present, and be released into the synapse, that is going to drive more neurotransmitter to be released into the synaptic cleft. So, in other words, what we have is, now if we modify that, if we additionally have another differential equation, let us repeat the dNt dt equals 1 minus nt divided by tau r. This is the recovery time constant, and our usage is based on the sum over j delta t minus tj times pt times nt. So, given that we have certain concentration of nt present, multiplied by the probability is going to be the one that is released for each and every spike, and that is how the nt is being reduced. Now, in terms of the idea of PT be reflecting calcium, we essentially need to make PT, we need to have a dynamics of PT, that is the dpt dt So, we will have to have a baseline probability of release. When there is no activity going on, then there is a certain probability of release, that is P0. So, P0 is baseline release probability. And so, that is what the probability of release will try to reach with a facilitatory time constant, let us say tau f. This is this is the opposite of the facilitation actually. So, PT is trying to go down to P naught if it is facilitatory, and tau f is the time constant that is pulling the probability of release to the baseline probability of release. And now the thing is that with we need a jump in the probability of release for every presynaptic spike, essentially reflecting the build up of calcium. So, that is given by J. So, that is the probability of release delta T minus Tj. There is a factor that we can put in AP times 1 minus Pt. So, that is the maximum the probability can go up to is our Pt, and for every time there is a jump proportional to the possible probability increase, and that is how the Pt is being modulated with every spike. And so, with the proper sign of AP, or we can I mean this AP is not provided as a as a I mean a particularly a negative number here, because we want a general model, because we can have this same thing in the short term depression also by changing the AP, or we can even make AP 0 with no change in probability of release, that is PT would be constantly at P0. So, depending on the situation, we can have AP represent different things. So, now as you can see that if the probability of release with every delta, every spike is kept on increasing, then in subsequent spikes there will be a larger probability of release, which will be reflected here, and a larger NT release, a larger reduction in NT. And so, this NT is also going to be the probability T times NT is going to be finally what is seen by the postsynaptic side, and with may be a factor that is, let us call it AD for now, that finally is reflected in the postsynaptic current due to the neurotransmitter. It is not that all the neurotransmitter is going to drive a current. So this can be representative of the postsynaptic neurons conductance G as a function of time, that is postsynaptic neurons synaptic conductance. So since this is a fraction, the maximum conductance can be taken as Gm, and it is AD times PT times NT is going to be our GT, and that conductance is going to be what we will take as driving the current based on the voltage change in the postsynaptic side, just like we have done earlier. And simply there also will be a equilibrium potential in the given the equilibrium potential represented as a battery in the branch, that is the synaptic current, and that can be that is based on the ions that is being transported, or rather ions that are being allowed to go out or into the postsynaptic neuron by the synapse. So essentially what we are saying is that we have our postsynaptic neuron, instead of an I external current, I mean this can also be there for, let's say, our patch clamp and so on. This is the capacitor, then if this is the sodium channel, this is the potassium channel branch. So this is ENA, EK, GNA as a function of time, GK as a function of time. Then let us say we have G leak, we have E leak, and we can have a G synapse, GS, and similarly an equilibrium potential. This was E leak, this we will call E syn in this side, and this GS, whenever there is a spike, would become it will follow the whenever there is a spike after a resting stage, it will produce a current that will be driven into this circuit. So it is sort of acting like almost like a source. In the other words, if we make the conductance equal to the this term here that we have put, and accordingly put in PT and NT and drive the system now with our summation delta T minus Tj with a finite width. I wouldn't want you to confuse this delta T minus Tj as a Dirac delta function, but just a very narrow pulse. So now with this we have connected what we had learned in the very first section of our course in computational neuroscience, where we were talking of models of neurons, and here with the explaining action potentials of the postsynaptic sides with GK and GNA and GL and capacitor, and we had assumed that a synaptic current is simply an I external. Here we have explicitly put in the synaptic current, and we still have an I external for an additional component that may be driving it. So that is the other one. So here we are including only one particular synapse in the model. So there may be many other types of synapses that still is reflected in the I external, or if we are doing a patch clamp, that would be reflected in the I external. So as with this AP or the jump in the probability of release, we have achieved the facilitation part, and we have also shown that with the same model we can do depression. So, whatever we have so far, that can still be modified further to bring in the 3-state model and still show facilitation. So, implementing these differential equations numerically is not that challenging in the sense that we can do this by simple integration of over small time windows, that is over millisecond or 1/10 of a millisecond, and directly make the derivative, compute the derivative at each point and find the next point and simulate these equations, because our time constants are not varying with the simulation. So, we do not need stiff solvers in order to simulate these differential equations. So far we have been talking about the short term plasticity from the point of view only of the presynaptic side. So, we have talked about neurotransmitter depletion, we have talked about calcium buildup and hence the modulation of probability of release. Similarly, these can be further extended if our P0, the baseline probability, that is also varying over time, which is a possibility in real neurons, that it is becoming more excitable with time because of plasticity, and this P0 may be varying with time over longer time constants. That can also be included in a very similar manner with a similar differential equation, and it is activity dependent, and it can be made activity dependent by making the differential equation as with a function of the summation delta t minus tj's. We can further have activity dependent replenishment, that is essentially in the previous model that we talked about, the tau r has a dynamics. So, basically we have a differential equation with d tau r dt, and tau r is now a function of time, and this tau r varies based on activity, and there is a lot of evidence about how with very strong activity the replenishment actually becomes faster, and the mechanisms of them are moderately understood. There are many more experiments to be done, and so here I mean we are trying to inform you about the possible complications or rather complexities that can be put into the presynaptic terminal and still be able to realistically model our short term depression and short term facilitation. So, all these actually play a role, and in our modeling work, in our computational neuroscience, when you actually go and use these things in the real synaptic data and so on, there you have to decide on how complex you want your model to be based on the questions that you want to answer, and that is true for even the Hodgkin-Huxley type of single neuron model also. So, you can have multiple types of channels, and make the model more and more complicated, but those complexities may be required for particular questions that you may be asking, and so that becomes the important issue. So, So far, all that we have done for the short term plasticity are presynaptic changes, I mean factors on the presynaptic side. So, there is one more important thing on the postsynaptic side for short term plasticity. And that is our receptor desensitization. So, this receptor desensitization essentially means that the postsynaptic receptors are become less and less sensitive to glutamate, or I mean not glutamate necessarily, to the neurotransmitter. I mean mainly studied with glutamate and AMPA receptors uh, because of overstimulation. So, the more and more activity there is in the presynaptic side, the more the receptors on the postsynaptic side get uh, desensitized. That is, they are not as much responsive as before. That is, their probability of gating reduces, keeps on reducing, and may go to 0. So, that also leads to a factor in the short term plasticity, and even if the mechanisms in the presynaptic side do not um, conform to short term plasticity for a particular synapse, based on simply the postsynaptic side's receptor desensitization, we could see short term plasticity. What I mean is that as we have discussed before, there can be synapses where the presynaptic mechanism, I mean whatever is going on in the presynaptic sides, is not producing the short term depression, uh, but we might still be able to see short term depression because of postsynaptic factors, and the primary among them is the receptor desensitization. If so, again we extend the same kind of differential equations to model that, and here we will say that dt is the pool of non desensitized receptors. So, let us go to the other page. So, here what we will have is dt equals, so the dt is trying to non desensitized, this is trying to go to 1 minus dt by tau d. This is the um, time constant for the uh, desensitized receptors to become non desensitized. That is, it is trying to get back its sensitivity. So, that is increasing the pool of non desensitized receptors, and with every presynaptic spike we are getting some factor of them being desensitized, and that is, we will have a decrease in the non desensitized population based on the activity in the presynaptic side, which again we will show by delta t minus tj. And now, how much is it used? We have had the discussion before, and that is this A d times P t times N t. So, that simply here is multiplied by A d times P t times N t, and now this will be multiplied by the non desensitized neurotransmitter population, receptor population. So, now you can see that we have the factor for facilitation, we have the factor for neurotransmitter depletion, we have a factor for the desensitization of neurotransmitter, all of them included in the model. And so based on our requirements and questions, we can choose the level of factors that we want to include, or the which factors we want to include, and accordingly model the synaptic transmission that will reflect short term plasticity, that is either depression or facilitation, or even if it requires to be constant for periods of time, that also can be done by modulating the baseline probability of release accordingly, or even bringing in dynamics of these factors also, which can also be happening. So with this we come to the conclusion about our discussions of short term plasticity, and we will later on be discussing along with other things the implications of both long term and short term plasticity, and we will be picking this up once more.